Now let's begin. Mitosis. Okay. We're about to talk about mitosis and the different stages of mitosis, of the different phases that mitosis go through. Okay. We also know that mitosis is somatic cells. Okay. Mitosis is somatic cells, so it's not dealing with our sex cells, but it's dealing with our body cells. Okay. But we're going to go through the different phases. All right. Remember, the abbreviation for those different phases are called PMAT. All right. PMAT. P stands for prophase, metaphase, anaphase, telophase. But you also have to have interphase. Interphase is before the PMAT. So you can say IPMAT if you want, but interphase will always be before any of those phases. Okay. So it starts off by saying the time between cell division. Okay. So interphase, one of the things that happens in interphase is that cells undergoes DNA replication and growth. Okay. And this is the time between cell division before it happens. Before cell division happens, this happens. Okay, so cell growth has to happen first before cell division starts, which is the PMAT. All right. The next thing we have is the cell spends most of its time in interphase. Yes, it does. Okay. In interphase, remember, we're dealing with multiple things. For example, you're dealing with cell growth, and you're also dealing with DNA replication. All right. DNA replication meaning the DNA doubles. Okay, it doubles. It doubles from 23 to 46. Okay, and remember, we get those cells. We get 23 cells from our mother and father. Okay, but this is what happens in interphase. Okay, cell growth and DNA replication, and get ready for PMAT. It's not getting ready for prophase, metaphase, anaphase, telophase yet, but it's getting ready. Okay. So it's not quite prophase just yet, but it's getting ready for prophase after DNA replication. Okay. There are 3 stages in interphase. Okay. G1 phase. Cells carry out metabolic activities to prepare for the S phase. All right. Remember, these are also checkpoints too. So as I move my cursor around here, I'm also showing you checkpoints. Here's the checkpoint right here. All right. This checkpoint is just basically saying that we're going to get, the cell, after cell growth, basically, we're going to get ready to go into S phase. But before we go through S phase, we want to make sure and check on the components. All right. Check to make sure everything is good. Okay. Before we go to the next phase. Why? Is because we don't want any errors. A lot of times when there's errors in cell division or the beginning stages of, cell growth, along with S phase and preparation for mitosis, things can happen in between those different checkpoints, all right, or different stages. Things can happen, all right, such as the S phase is not double up, okay, from 23 to 46, it doesn't double up, so it can occur some genetic dysfactors can happen between that. All right, you have the S phase, says synthesis phase, okay, DNA is replicated, all right, again from 23 to 46. G2 phase, organelles and molecules required for cell division are produced. Okay, cells prepare for mitosis. Okay, so the organelles, which is the cell parts, which we talked about. For example, the vacuole, the Golgi bladder, what else? Let's see, the vacuole, the mitochondria. Okay, those are all cell organelle parts. All right, that are beginning to form inside of the cell. All right, but this is all during interphase. All right, before we prepare for mitosis, before the split. All right, let's move on. All right, now here's an example of interphase inside of an animal and plant cell. Okay. Cell division phase, mitosis. Okay. Again, preparation for mitosis. Not quite mitosis just yet, but it's going through preparation. All right. Okay, mitosis says process whereby a cell will divide to produce 2 new identical cells. All right, I didn't say different cells, identical. Okay, the cells are the same, 2 daughter cells. Both of those cells have a pair of each other. Okay. Next, allows organisms to grow and replace old, damaged, or dead cells. Okay, now this is one of the benefits. These are some benefits of mitosis. Okay, so some benefits of mitosis basically can replace old cells, also damaged cells as well, or dead cells. Okay, and this comes from basically from a cut or some type of fracture or dislocation. Our cells can pretty much go through the process of a healing, and mitosis is a way of our cells healing or multiplying enough to recover from that damage. Last thing, occurs in all body cells. Okay, so another word for body cells is somatic cells. Somatic and body cells is the same. Okay, so somatic would be somatic cells. Okay, so anytime you see somatic cells, just understand that those are body cells, not sex cells, but body cells. All right. Let's see. Parent cell we have here. If you look at my cursor, just follow my cursor. All right. These are the parent. This is the parent cell, the original cell, one cell. Then it goes through DNA replication, okay, which happens in the middle of S phase. Not S phase, but in the middle of interphase. All right, S phase is another word for DNA replication too, but it happens in interphase, the middle. All right, we have chromosomes are copied and doubled in number, okay, from 23 to 46. All right, chromosomes now split, okay, so they're actually getting ready to split. As you can see, there's a red on this side, there's a blue on this side, there's a red on this side, there's a blue on this side. That's because the chromosomes start to split from one another, okay? And then the last step, we have cell division, okay? We have cell division. There are two daughter cells, but before that step, cytokinesis usually happens. Okay, cytokinesis, well, actually not usually, but it does happen. So cytokinesis actually happens before these two identical daughter cells split. Okay, so every cell in your body contains the same genes, but only some act to make the cell specialized. All right, for example, nerve or your muscle tissue. Okay, prophase. All right, prophase is the first and longest phase of mitosis. Yes, it is. Spindle fibers. All right. Remember, I told you we're going to call these things Twizzlers. Okay, just to remind you, we're going to call these things Twizzlers. All right. And we said that these are the Jolly Ranchers. All right, but the science vocabulary term for these type of structures are actually called centrioles. Okay. Strands of DNA condense and thicken to form visible duplicated chromosomes, sister chromatids. Okay, which are one of these guys here. Only one, not two, but one. Okay, and they form into these. Remember, before the DNA, all right, before the DNA formed these chromosomes, it was basically a lot of scribble. Okay, it was a lot of scribble to those chromosomes basically compacted with each other and formed somewhat like an X shape. Okay, but they're one by itself are called sister chromatids. Sister chromatids are held together by centromeres. Okay, centromeres. These little dots here that's in the middle of these colors, those are called centromeres. All right, and they hold those chromosomes together. Okay, the nuclear membrane breaks down. Okay, eventually this nuclear membrane right here. All right, as you see my cursor, not this membrane. I'm talking about this one, not this membrane, but the purple dotted lines that you see. These are going to disappear. Okay, they're going to disappear. All right, again, we're still on prophase. Again, these are called the spindle fibers. These are the Jolly Ranchers, all right, which in other words are centrioles. Okay, the centrioles move to the opposite poles of the cell. So these centrioles and spindle fibers are actually moving away from each other, not together, but they're actually moving away from each other to opposite poles. All right, in other words, we're getting prepared for metaphase. Okay, but again, what hold these sister chromatids together are the centromeres, and together they are called the sister chromatids are called chromosomes. All right, paired chromatids. Okay, spindle fibers begin to form from the centrioles. Chromatids become visible under the light microscope. Okay, so before prophase, you really can't see these chromatids under a microscope. All right, you can't really see them. It's still kind of too small. And not only that, but the there's not really a shape just yet. Okay, you really start to see the shape once they form into chromosomes. All right, but when that DNA is just kind of scribbled, it is not compacted to form these types of shapes. A lot of times you just can't see it. Okay. All right, now this is an example of prophase. Okay, we have an animal cell and a plant cell. All right, again, the spindle fibers are what we call the Twizzlers, and the centrioles are what we call Jolly Ranchers. Remember, those centrioles hold the spinal fibers. All right, spinal fibers, which is the Twizzlers. All right, now we're on metaphase. Okay, now it says the second phase of mitosis. Spindle fibers connect the centromere of each chromosome to the poles of the spindle. Okay, so it kind of all depends on how many chromosomes, the amount of chromosomes that are in the cell, that you can, really see a variety of spindle fibers that's coming out of the centrioles to attach to the centromere. Okay, but as you can see in this picture, we have yellow, blue, green, and red. Okay, if you look right here where my mouse is actually moving, I'm actually got my mouse on the spindle fibers, the Twizzlers, that are coming down to attach themselves to the centromere, all right, that hold the sister chromatids together. It's coming there. All it's going to do is basically pull that chromosome apart. It's going to pull them apart, okay? Spindle fibers connect the centromere of each chromosome to the poles of the spindle. Spindle fibers help chromosomes line up across the equator center of the cell, okay? So whenever you see the chromosomes line up in the center of the cell, as you see here, all right, just understand that that phase or that position is basically representing metaphase. Okay, metaphase. When the chromosomes line up, okay, they line up. All right, and basically what I'm saying to that is eventually those chromosomes are going to be pulled to become sister chromatids on each side. Okay, on each side. Spindle fibers help chromosomes line up across the chromosomes and line across the equator center of the cell. And the equator center of the cell will actually be the middle. It will actually be between this blue and this green. Okay, between these here. That is basically the equator line. All right.